And we're back. Let's move on to understanding number 6. The genome is the whole of the genetic information of an organism. So, a genome is the entirety of an organism's genetic information. That includes all sections of DNA which are found in chromosomes. Here you see a picture called a karyotype. A karyotype is an image of an individual's chromosomes. It looks like it's cut off a little bit. Yep, because our chromosomes 19 through the rest are cut off. But we'll talk more about that when we actually talk about chromosomes in 3.2. Some of you may remember from freshman year when you did the karyotype posters where you looked at all the different genetic disorders that result from missing chromosomes or extra chromosomes or pieces missing. That was a lot of fun. Understanding number 7. The entire base sequence of human genes was sequenced in the Human Genome Project. So the Human Genome Project began in 1990, and it aimed to find the DNA sequence of the human genome. So a minute ago, I said that a genome was all of the DNA contained in an organism, right? Not necessarily an individual organism. It can be, so like your genome, right? But most of the time we talk about across an entire species. And when you're talking about across an entire species, that means that genome contains all versions of all genes in that species. So humans have approximately 23,000 genes, which is a lot less than we expected. Most of the DNA in our genome is not expressed, right? So it's not like that. Not every single piece of DNA in that long strand that makes up all of your DNA, it results in a protein. There's lots of sections that have regulatory functions. So they are in charge of regulating the rest of the DNA. They're in charge of regulating how much of a gene is expressed. So say you have the gene that produces insulin, right? There are other genes that control how much transcription and translation happen to produce that insulin. So you can think of your entire genome as a giant switchboard. And you don't just have the switches that make stuff, you have other switches that turn those switches on and off. So it's a pretty complicated system. So let's actually compare the number of genes in humans with other species to see how we stack up. So species have a very wide range of gene numbers, and the number of genes that you have is not reflective of an organism's perceived complexity. And you can see that in this chart. So if you look at influenza, which is technically a virus, right? It has about 11 genes. Now, E. coli, which is a bacteria, much more complicated, right? It has over 4,000. Then you have a fruit fly, which has about 15,000. And you'll notice that these numbers tend to be relatively small compared to other ones, and that's why we use these particular species as models when we're doing investigations, because we have a limited number of genes, we can control the process. Chickens, 16,000, humans, 22, grapes, 30,000. Now, people are like always astonished that this is the case. I am not surprised at all. And those of you that know me know I have a mild to moderate passion for plants. I think they basically rule the world, and I think they're way more interesting than animals will ever be on any given day. And that's because they can do so many dang things. Remember, if all your genes are like a deck of cards, they have so many more cards than we do. They can do so many more different things. Biochemically speaking, think about all the genes that it requires to be able to do photosynthesis, all those extra proteins that you have to create, all those extra metabolic pathways, right? So it's not crazy to me that they have like legit way more genes than we do, because they can do things that we can't. They can synthesize amino acids. We can't. They can photosynthesize. We can't. We have to like move around and find our food. They make their own dang food. Think of all the different chemical pigments that they make for their leaves and their flowers. All the different things that they can do. So yeah, it makes sense to me that they have way more genes than we do. Skill number 1. Use of a database to determine differences in the base sequence of a gene in two species. Now we're going to do an activity with this next rotation, but let's just talk about it now anyway. So as the genomes of more species are sequenced, online databases can be used to determine the sequence differences between species. The following table shows nucleotide differences in cytochrome C genes. So cytochrome C, let's see if I can remember what cytochrome C does. Cytochrome, so cell, chrome, let's see. I want to say it has something to do with oxygenation within the cell. I'm not sure. I can't remember off the top of my head, but that's okay. So let's just take a look. So you'll see we have a table and we have species going down. We have species going down and we have species going across, right? And you'll notice we have human, rat, mouse, chicken, fruit fly, yeast, rat cytochrome, mouse cytochrome, fruit. Okay, yeah. So same thing going across. All right, so these are all eukaryotes. So this has got to be like a eukaryotic thing. And I'm guessing it's an aerobic respiration thing. Just my guess. And you'll notice these same species are across the top. And what you can use this matrix, that's what it is, what you can use this matrix to look at the differences in the number of base pairs. So I'll show you how this works. So if we're talking about one gene, it's the cytochrome C gene, right? And we're not going to look at the actual gene because like who wants to do that? We're just looking at the number of differences. So if I want to compare humans, if I compare humans to humans, I'm going to have 0 differences, right? Because they're both humans. But if I compare humans to rats, that means there's 34 differences, 34 different base pairs in the DNA of the cytochrome C gene between humans and rats. If I wanted to compare chickens to yeast, okay, so here's chickens. Now I go all the way over to yeast. Right? There you go. 119 differences. Now, you can use this to help you figure out who is more closely related in terms of evolution to who. Because the more similar they are, you would expect the more closely related they are. Now, you can't just do that for one gene, right? Because some genes are kind of universal to all living things because they do really, really important things. And there's not much change because they're good at what they do. So you can't just do it with one gene. You have to do it with like a whole bunch of genes. And then you can get like, if your genome is most similar to another species' genome, you should be more closely related. So if you think about chimpanzees and humans, they are 98% the same. That means there's only 2% difference in DNA between chimpanzees and us. Now, 2% doesn't sound like a whole lot, right? But clearly, it makes a difference. Okay, so let's see. If you were looking at this chart and you had to find the smallest number of differences, so what 2 species in this chart are most similar in terms of their cytochrome C protein? If you said mice and rats, you are correct. And that makes sense because mice and rats look like they're pretty similar, right? If you had to figure out who was the most different, who has the most differences in their cytochrome C gene and protein, let's see, who would it be? What's the biggest number on the chart? 123? Yep, I see a bunch of 123. So here's a 123, 123, 123. Oh no, here we go, 133. So who is it? Let's see. Yeast and fruit flies are the most different, apparently. Interesting. So that's how you would use a table like this to compare differences between genes in different species. Same genes, different species. Okay. At this point, you should be able to do the following things. You should be able to define the terms gene, locus, genome, allele, and mutation. You should be able to outline the mutation that caused sickle cell anemia. You should be able to describe the Human Genome Project. And you should be able to compare the number of genes found in humans and other species. Once you've done all that, you can move on to the next part of this assignment. Make sure you upload your notes per usual. However, there's more. So if you go into Canvas, you will see there are 3 discussions, genetic reflections. So let's see. We'll discard those. So let's see. Here we are in our modules. This is what we're doing right now. You'll see down here, look, Genetic Reflections Video 1, Genetic Reflections Video 2, Genetic Reflections Video 3. Now these are discussions, okay? So for each one, you're going to click on the link. And you're going to watch the video. And you're going to respond to this video with 3 posts. The first one is going to provide a question that you have about the content. The second one, you're going to respond to someone else's question with your own insight. And the third one, you're going to discuss your own reaction to the video. So does this affect you or might it in the future? What do you think? Okay? And you can start replying down here. When you're done with that one, you're going to do the next one, which is how CRISPR lets us edit our DNA. And this is a TED Talk by Jennifer Doudna. And you're going to do the same thing, right? Same thing. Provide one question you have about the content, respond to someone else's question with your own insight, and then discuss your own reaction to the video. Now, these are graded discussions. They're 5 points each, so make sure you do all 3 posts. And there's one more. So what's the next one? Okay, here's the last one, right? Should you be able to patent a human gene? Interesting, right? I can see pros and cons on both sides. Think about genetic medicine. So yeah, that is the rest of your assignment for today. Next rotation, we're going to do a little bit of work on that gene database stuff. And I think Zane might have a video for you guys to watch too. But that's all for now. As always, miss you guys. And looking forward to looking at your responses to these videos. I expect to see some lively exchange. And both classes, you're all grouped together now, so you don't necessarily have to respond to someone in your section. You can respond to anybody. Okay? All right, have fun. See you guys later.